Hello, my name is David Schreiber. I'm a professor and chair of the Department of Biomedical Engineering here at Rutgers, and I'm happy to give you this walking tour of our department. Along the way, I'll tell you a little bit about our history, our facilities, our training, and our research. The Rutgers Department of Biomedical Engineering is located on Bush campus in Piscataway, New Jersey. As you may know, New Jersey is one of the leading locations for the pharmaceutical, biotechnology, and medical device industries, and the proximity of these industries is a major reason why our program is successful. The BME building is the home to our program. Having opened in 2007, this 80,000 square foot building houses our classrooms, teaching labs, research labs and facilities, and faculty, graduate student, and administrative offices. The atrium of the building is a place for students and faculty to congregate between classes and before and after seminars. The openness of the atrium is also symbolic of our open and collaborative culture. From the atrium, you can see up to the second and third floors. There is also a basement which includes laboratories and specialized research facilities. Adjacent to the atrium is a 140-person auditorium. Research seminars and many of the core BME classes are held in this auditorium, which is fully equipped with the latest audiovisual technology. At Rutgers, engineers join their major department in their second year. BME is currently the third largest engineering major, with class sizes of around 125 to 140 students per year. All of our core lecture classes are taught in multiple semesters, so most classes are capped at about 75 students. The primary exceptions are lab classes, which are taught in only one semester. These classes typically break out into sections of 20 to 25 students each. The first floor is primarily dedicated to instruction and BME administration. In addition to the auditorium, the first floor has a mid-sized classroom, which you can see here, a modular classroom, and two smaller classrooms for teaching electives. The first floor also has a computer lab and 3 teaching labs. The computer lab includes over 20 PC workstations that include specialized software used in instruction. The lab is open for BME students to use during the day, except when it is being used for class. Several of our faculty specialize in computational research. For example, Professor Yannis Androulakis studies systems biology using big data. He analyzes a variety of omics data, such as genomics, proteomics, and metabolomics, to understand how our body clock or circadian rhythms help to control our own immune system. He applies this to personalized medicine. This research can help define what kind of medications work best on a specific person and when the right time is for that person to take their medicine. Dr. Lee Tsai examines gene expression data to understand neural stem cell populations. He applies this to improving regeneration after spinal cord injury and to understand how our nervous system develops. Dr. Thomas Papathomas conducts fascinating research into optical illusions and tries to understand how and why what we perceive can be different from the reality. Dr. Troy Shinbrat studies complex problems in fluid and solid mixing. He applies principles of transport and reaction kinetics to study problems in pattern formation, such as how and why folds in our brain occur. Finally, Dr. Ilker Hajialioglu uses principles of machine learning to develop approaches for image-guided surgery. His emphasis is on ultrasound imaging, which is a low-cost, portable imaging option. He works to extract important information from ultrasound images that can be used to guide procedures in surgical biopsies and orthopedic surgery. One of our teaching labs is focused on biomechanics. This lab includes equipment to characterize how stiff and strong natural and man-made materials are, and to look at aspects of flow and transport. Each lab section typically includes 20 to 25 students. There are several faculty who specialize in biomechanics research. Professor Bill Cralius is one of the pioneers in actuatable prosthetics. In Dr. Cralius's design, a lower arm amputee can control the movement of prosthetic fingers by flexing muscles in the upper arm. Professor Noshir Langrana is one of the leading experts in spinal biomechanics. He has used a variety of experimental and computational tools to understand spinal fracture and intervertebral disc disease, and has worked with clinicians on spinal devices. The next teaching laboratory is for systems, devices, and instrumentation. In this lab, students study the electrophysiological signals that are produced by the body and learn how to acquire, filter, and analyze these signals. Simulations are also used to demonstrate clinically relevant circumstances. Many faculty focus on devices and instrumentation for their research. Professors Gary Drzwicki and John Lee study cardiovascular engineering. They design approaches, devices, and models to collect data on things like blood pressure, heart rate, blood flow, and vascular tone, and use that data to design possible approaches for interventions. Professor George Schoen studies the control of eye movements and has applied this work to aspects of sports, such as putting in golf and shooting free throws in basketball. The final teaching laboratory is for what we call wet lab experiments. These labs are for training in techniques to understand cells. Students can learn how to culture cells, image them with microscopes, and measure protein expression. Research with cells in culture is prominent among our faculty. Professor Martin Yarmish runs a multifaceted research program that especially emphasizes regenerative medicine. One aspect of his program is to encapsulate stem cells and use them to treat different inflammatory conditions. Dr. Bijou Parakadin encapsulates cells with an eye towards biomanufacturing for immunoengineering. Dr. Parakadin has also developed exciting technology for the rapid cloning of large fragments of DNA. Many of our faculty have a particular focus on neuroengineering. Professor David Schreiber is designing a drug delivery platform that targets drug payload based on the production of free radicals, which are elevated following brain injury. He is also an expert in the study of how axons, which connect one neuron to the next neuron or end target, are damaged during trauma. Professor Maribel Vasquez develops microsystems to understand how neurons are guided to grow and regenerate, with particular emphasis on the visual system. By understanding how retinal neurons are guided in these microsystems, she hopes to design therapeutic interventions to restore and improve vision in vivo and ultimately in patients. A final laboratory on the first floor is the home of our Medical Device Development Center, or MDDC. Professor Kristen Labazo heads these efforts, which includes a partnership with the Matheny Hospital and Medical Education Center, which is a local facility that specializes in caring for severely disabled patients. Our students can spend time at this facility over the summer and observe interactions among patients, loved ones, and practitioners. The goal of these observations is to identify areas of needs where a biomedical engineer can intervene. This could be modifications to a wheelchair to improve range of motion for a patient, or a more efficient and convenient way for nurses to sterilize wheelchairs, such as with a blanket decorated with UV LEDs to allow the nurses to spend more time with patients. The students then build their prototype devices in the MDDC. The MDDC includes several pieces of makerspace equipment, such as 3D printers and laser cutters. These have also been used by Rutgers faculty, especially Professor Jay Tsai, to 3D print personal protective equipment, or PPE, for frontline healthcare workers during this COVID-19 pandemic. We've seen here headbands for face shields being 3D printed. The MDDC is also where one of our coolest pieces of technology is being developed by Martin Yarmish in his laboratory. This is an automated venipuncture device, which is designed to allow hands-off intravenous blood draws. The device works by using near-infrared imaging to light up your hemoglobin so that an appropriate vein can be identified. Using artificial intelligence, a needle is then guided using ultrasound images to be inserted with a 9-degree of freedom motor that can move the needle with the vein if there is slight patient movement or if there is rolling veins. The lab is now developing a handheld version for ambulatory and bedside care, as well as technology to immediately analyze the blood for a complete blood count or other common tests. The second and third floors house the majority of the lab space, as well as graduate student and postdoc cubicles and faculty offices. As we look down this hallway, cubicles are on the left, and to the left of that are faculty offices, and labs are on the right. Here we see equipment for the high throughput rapid synthesis and characterization of different synthetic polymers and polymer-biopolymer hybrids. Polymer synthesis typically must be performed in highly controlled environments, but Professor Adam Gormley has discovered approaches to control synthesis on a regular lab benchtop in open air. This has greatly simplified the process, and now Dr. Gormley is rapidly producing large numbers of different polymers in parallel using automated robot technology to characterize the polymers and machine learning to make adjustments to these synthesis protocols on the fly. Dr. Gormley's goal with these polymers is to rapidly and efficiently converge on designs that mimic the structure and activity of natural molecules, such as cytokines and growth factors, which are otherwise very expensive to produce and purify. Professor Francois Berthiaume is also developing nanotechnology, this to deliver hemoglobin to healing wounds to improve oxygenation and vascularization. In the field of drug delivery, Dr. Charles Roth has developed multifunctional polymers called graplons as a delivery platform that can enable self-assembly with peptides or oligonucleotides into nanoparticles, can protect the drug from early clearance, improve cellular uptake, and have a pH-sensitive background for smart release of cargoes into tumors. Dr. Jay Tsai is developing technology to improve gene delivery to control protein expression in the brain, as well as efficient means of controlling delivery and maintaining appropriate concentrations through the cerebrospinal fluid, or CSF. As I mentioned, each floor also has a series of smaller rooms with specialized equipment. Here we see a microscope suite that includes a confocal microscope, which is used to generate clear images in 3 dimensions. We have faculty who specialize in imaging for their research. Professor Nadu Bustani uses optical techniques to relate the biophysical properties of cells to their biological and functional properties, including, for example, how force is transmitted as information to a cell, and what biophysical changes in organelles occur during apoptosis, which is a key cellular regulator to prevent cancer. Dr. Mark Pierce uses different imaging modalities to perform cell and tissue diagnostics. One of his areas of interest is global health. Dr. Pierce is designing low-cost optics that can be connected to a cell phone camera to conduct virtual biopsies in regions of the world that have poor access to healthcare. The 3rd floor is similar to the 2nd floor in layout, with some special facilities and equipment that I would like to highlight. The BME microfabrication cleanroom is on the 3rd floor, where microdevices and lab-on-a-chip technology can be developed. This facility allows for the fabrication of devices with micron-sized channels for the controlled mixing of fluids and the deposition of micron-sized electrodes to deliver and record electrical signals. Professor Jeffrey Zahn directs this facility and leads our efforts in creating micro-electromechanical devices, or MEMS devices, for biomedical applications. Professor Zahn, with collaborators, is developing ultra-flexible probes for inserting into the brain to record electrical impulses for brain-computer interfaces. He is creating on-chip devices to study brain and spinal cord injury, and he is developing devices to understand the differences between neurons generated from cells from the skin of control patients and those with a neurological disease or disorder, such as autism, drug addiction, or schizophrenia. Finally, he's creating miniature devices to reliably deliver DNA and other molecules to cells using electric fields. Much of the research on the 3rd floor is focused on biomaterials. One interesting set of equipment includes this glove box and electrospitting setup. Here, a polymer in solution is extruded through a syringe while experiencing a strong electric field. The field draws the polymer to a collection plate while the solvent rapidly evaporates. The result is a fine thread that can range from tens of nanometers to microns in diameter. These polymer threads often mimic the size and strength of natural biofibers and can be useful in tissue engineering. For example, Professor Joseph Freeman loads this contraption here with the electrospun fiber, which then braids the fibers into a strong weave that can be used someday for tendons and ligament replacements. Dr. Freeman is also working on bone tissue engineering and 3D printing scaffolds that mimic the structure of bone to hopefully allow for the proper regeneration of bone function. Dr. Adrian Mann studies the molecular signatures of different materials to understand problems in bone formation and tooth decay. Finally, Professor Prabhas Moge develops nanomaterials and nanostructures for delivering therapeutic cells and molecules to the brain. He particularly focuses on delivery of antioxidants as well as stem cells, which can regenerate lost populations of cells in diseases such as Parkinson's disease. Thank you for taking this virtual tour of our program. I hope you found it informative and perhaps inspiring. Biomedical engineering is a wonderful and exciting field where people can use their engineering skill sets to understand the mysteries of life and directly impact human health and welfare. And Rutgers is a fantastic place to learn and practice biomedical engineering. For more information, I encourage you to visit our website and to follow us on Twitter.